Well, thank you for everybody for joining us back after the break. We have two more exciting speakers. We're going to shift gears a little bit. We've heard a lot about administrative databases, retrospective analysis of EMRs, hearing about how sepsis influences the economy of our country and our province. Next, we're going to shift to more discovery type work, which relates to understanding the biology of sepsis. And here we have two interesting speakers. The first is Dr. Evan Haney, Haney. I hope I'm pronouncing that right, Evan, who I just got a chance to meet a few minutes. I just got a chance to meet a few minutes ago. He's the Chief Scientific Officer of ASAP Medical. He works together with one of the pioneers of sepsis discoveries related to microbiological determinants and prognostic determinants of sepsis, Dr. Bob Hancock at the University of British Columbia. And here today he will talk to us about novel sepsis diagnostic assays to help risk assess patients for progression to severe sepsis. And his talk is entitled Advances in Sepsis Diagnostics and Biomarkers. Thank you.
All right, so thank you very much, everybody. I'm going to put this up a little bit. Everybody can hear me? If I have to bend over, I guess I'll do that. So thank you very much for the invitation to come and speak with you today. Oh, perfect. Here we go. I do work with Bob Hancock at UBC, and I'm also the CSO of Sepsis Biosciences, which is a subsidiary of ASAP Medical. My conflict of interest is we are developing a sepsis diagnostic, so I will say that up front. Bob sends his regards. He is actually in Australia right now visiting his grandchildren, so that's why you are all getting to have me here today. So I'm going to give a very quick introduction to sepsis. This crowd doesn't really need it. We've already had some really great introductions on that earlier this afternoon. Obviously sepsis is a huge burden globally and causes a massive number of cases, huge number of deaths. And as we've already kind of talked about today, it's really hard to be able to diagnose patients because some of the early signs and symptoms of sepsis are not really specific. One thing that I will point out is sort of the progression, the immune response of sepsis. So sepsis is characterized by this, I'm hoping I've got my laser pointer here. So sepsis is characterized kind of by an early hyperinflammatory phase or a so-called cytokine storm. And then over time, these patients will go on to more of a hypoinflammatory phase or an immune suppressed phase. Originally, a lot of work focused on this initial cytokine storm, and it was thought that this sort of burst of hyperinflammation was what was responsible for a lot of the immune response and the negative aspects of sepsis. But now this hypoinflammatory phase is actually becoming increasingly appreciated for its relationship to sepsis and long-term outcomes. And the only reason I'm highlighting this is a lot of our technology is actually based on kind of this idea that there's an underlying mechanism of what's going on at the cellular level that sort of dictates this switch from this hyperinflammation to a hypoinflammation. And so just really quickly, as I'm going to start here, some important considerations are what we'd like to see in a sepsis diagnostic test. So I think it's really useful to look at the definition of sepsis. So the sepsis 3 definition, as we've already seen today, is a life-threatening organ dysfunction caused by a dysregulated host response to an infection. So if we look at that definition, there's really two key components of it that we need to be looking at. First, you need to be able to look at the immune status of the patient, as well as figure out what is the pathogen or what's making them sick. So in particular, can you identify, is it a bacterial or a viral infection that's making them ill? And also whether or not their immune response is actually going to progress to this organ dysfunction. I think it's also it's been well shown today that sepsis is a medical emergency and that the faster that you can identify a patient, the better their outcomes down the road. And lastly, it's also important to look at both sensitivity and specificity when you're looking at a sepsis diagnostic. So first of all, we want to have a sensitive test so that we can correctly identify those patients who are at greatest risk of going on to severe sepsis. That way we could prioritize treatment for them. But in addition, having a high specificity for a test also allows you to identify those patients at low risk of sepsis. And then that way you can prevent maybe using the empirical use of antibiotics. The overuse of antibiotics, which we already saw earlier today, is also a big problem in our healthcare setting, which is also contributing to antimicrobial resistance. So some current strategies that are used for diagnosing sepsis. So the first one is pathogen identification, and the gold standard, I think, is blood culture. The problem with blood culture techniques is they can take up to 2 days to get a result. And so they're actually quite slow, and in many cases you may not actually get a positive blood culture, but a patient may still go on to sepsis. There are other types of technology like multiplex PCR, mass spec that are bringing down the time required to get a result from these tests. And certainly knowing the pathogen that's making somebody sick is really important to be able to address that infection. But there's also the immune component of the sepsis that really is not appreciated by just knowing the pathogen that's there. There's various biomarkers that are available that people use to look at sepsis. So some of the ones that we've seen already are things like procalcitonin, C-reactive protein, lactate levels in patients are also measured. There's several others that have been proposed in the literature, so various people have seen different proteins sort of correlate with sepsis outcomes. I've also highlighted here the one that recently just got approved. It's a test called IntelliSep. It's actually based on the shape of your white blood cell, so I'm grouping it as a biomarker. It might be generously called that way. And some of these biomarkers work, but the problem is that they're not universally applied. And there's no they don't actually have the best sensitivity or specificity depending on which patient population you're looking at. So there are challenges still, even just looking at biomarkers. In addition, they may not be specific for sepsis. They may be reporting on another disease. Machine learning algorithms, so we had kind of a talk on this earlier today, would be taking clinical records or even results from some of these other biomarker assays and basically having a computer monitor those particular inputs and then automatically score patients on whether or not they're going to go on to severe sepsis or not. This could be happening in the background and would certainly could be a way to identify those patients in hospitals without having a physician needing to look at charts all the time and make that decision. Many of these are promising, but they're still in early days, and I think the talk earlier today also showed some of the challenges associated with those. The last one is looking at the host immune response, and this is the stuff that our work is interested in, but there's also several other companies and products that are out there on the market that are looking at this immune response component of of sepsis patients to be able to diagnose sepsis. So I'm only going to be focusing on the tests that we're developing at SEPSAT. So this work really started from a graduate student in Bob's group named Olga Pena. And what she did was early on in her PhD work, she was interested in looking at sepsis, and she was looking at this phenomenon called cellular reprogramming or endotoxin tolerance. So what she was able to do is take white blood cells from healthy patients and put them in a petri dish, and then basically trying to simulate what might happen under sepsis conditions. And so she would stimulate these white blood cells with a single dose of bacterial LPS, kind of to mimic a bacterial infection. And what she could see was you would have an expected pro-inflammatory response or an inflammatory response to this LPS. But the interesting thing that happened was if she then stimulated it again with a second stimulation of LPS, she then saw that these white blood cells became immune suppressed, kind of reminiscent of that trajectory that I showed you on the first slide of sepsis. So based on that in vitro observation, her and David Hancock, they went and looked in the literature at gene expression profiles among patient samples, which you can see over there on the left. Yes, left. I'm facing this way. Anyway, on the left. And what they found was that this gene expression profile that she was able to find in white blood cells stimulated with LPS, it actually showed up in sepsis patient samples, demonstrating that this cellular reprogramming phenomenon was present among sepsis patients. And indeed, if she took the signature, which is 99 genes, she could find that they were interrelated by a protein-protein interaction network, which you can see down there at the bottom, suggesting that this is also biologically relevant and mechanism-based for what's going on in these sepsis patients. In this study, they did a small clinical trial just to test and see whether or not they could recapitulate this in fresh samples from patients collected in the ER. And indeed, they were able to do that. They found that in blood collected when patients came into the emergency department, that this gene signature was upregulated in sepsis versus non-sepsis patients, and that it was also associated with organ failure. So based on that initial study, what we've gone on to do now is to perform a larger clinical study in 500 patients, and part of the reason for that was to be able to confirm this sepsis study and then also refine this sepsis severity gene signature down to something that would be useful in a diagnostic assay. The important thing to note about this clinical study is that this blood was collected in tubes when the patients first came into the emergency department. So we're really trying to capture their gene expression, that immune response when the patient first presents to the hospital. And then the outcome that we were looking at was their 24-hour SOFA score. So all of our modeling is based on their initial presentation in the hospital and then what happens 24 hours later. So through a lot of work, through our data scientists, this is just showing you how we've gone about developing this subset ER model and also moving it towards an RT-qPCR kit. So the first thing was collecting the data from that clinical study. There's some pre-processing that happens, and then we went and trained this model using these 514 patient samples, which contains a mixture of sepsis patients and non-sepsis patients. Through several rounds of model tuning and cross-validation and sort of optimizing the model, we were able to come up with a very good performing model, which I'll show you on the next slide. And then we were also able to apply that to now 23 data sets, which includes both published data sets and new clinical samples that we've collected along the way. And then we're now working on deploying the model and then migrating it over to an RT-qPCR platform that can be useful as a diagnostic test. So very quickly, here is some performance of how this gene signature works. So you can see in the top table is the performance of looking at patient samples either from the ICU or the ER, and you can see that we get very good sensitivity and specificity across the board. We perform really well against ICU patients, but we're still doing pretty well even amongst those patients who are coming into the ER, which are among the most challenging patients to deal with. I will point out too, we also did COVID-19 samples, and this includes both emergency room and ICU patients, and you can see that our AUC now is approaching what we see in the ICU samples. In addition, we've also, in collaboration with the group out of Toronto here, have done testing on digital drop PCR, which is akin to the RT-qPCR method we're going to be using, and we see that the test also performs well there. And then on the bottom, I'll just highlight that we've tested this against multiple other data sets that are in the literature, and it performs very well, better than other published gene expression signatures that are out there. And in addition, it's also specific for sepsis. So if we look at gene expression amongst cancer patients, we see that the model performs poorly, which you would expect because it shouldn't work on differentiating cancer versus no cancer. So I'm going to skip over that for the interest of time and just get to this. Give you an idea of what the test will look like when we actually get it ready to go. The test will be fairly simple. You would take a blood sample from a patient. You would isolate the RNA from that in a rapid protocol that we're working on developing. You then run the test kit on the RT-qPCR platform, which gives you the gene expression of the signature genes. It's 6 signature genes and 2 housekeeping genes in our case. You then take those relative expression values and plug it into the model, and this will return a risk assessment score for sepsis severity. And if all of these things work well, you can have this result in about an hour. So we've developed a prototype assay kit, and we are just at the process of initiating a pilot clinical study at St. Paul's to be able to look at this on sepsis patient samples. So in the last few minutes here, I'm going to shift gears a little bit to talk about where we think the future of sepsis diagnostics might go. And so I'm going to introduce this concept of endotypes. And an endotype, it can be considered a subtype or a phenotypic cluster of a condition. And it's defined by some sort of functional or mechanistic underpinning for what's going on in the patient. And so in terms of sepsis, you can imagine that as patients come into a hospital, sepsis isn't equal across everybody. There's likely multiple different types of patients that are coming in. And what endotypes might allow us to do is to be able to break those up into further mechanistic groups and see, okay, can we see among all patients who come in with sepsis, maybe we can identify those that are in a low mortality risk group, but then further look at those that are very high mortality risk, and then maybe even further divide those those high mortality risk ones into some sort of functional mechanistic reason why their sepsis is progressing. And then I think our big goal is that this could enable a mechanism-specific therapy for sepsis patients. So to get at this question, this was one of Bob's former grad students, Arjun Bagela, and he looked at transcriptional data from 348 patients and was able to actually identify 5 different endotypes among sepsis patient samples. And he indeed found that 2 of those were particularly associated with a poor prognosis for the patient, whereas 3 of them were involved in a better prognosis. And really exciting is that each of these different 5 endotypes, you could actually be able to classify a sepsis patient just based on the expression of 2 genes. So this now adds an additional layer of a diagnostic that could be applied to then further divide these patients up into these endotypes. And this has the potential to be developed as an endotype-specific test for sepsis. The other thing, so this was brought up in one of the talks earlier, was what is going on with sepsis in the COVID pandemic. So this was work from Andy Ann, another grad student of Bob's, and what he was looking at was the gene expression profile of patients as they went into the ICU. And he compared COVID-positive ICU patients to COVID-negative ICU patients. And he looked at it at 2 different time points. So he looked at a gene expression profile when they came into the ICU and then 7 days later. And what he found is that early on in their ICU stay, you could see that COVID-positive and COVID-negative patients were differentially expressed. So they had a difference in their gene expression signature, which you can see here based on the clustering of the differential clustering of the yellow and the purple on the PCA plots there, as well as this volcano plot, which shows you the genes that are differentially expressed between those 2 patients. And then when you look at what those genes are that are differentially expressed, you can see that a lot of them are related to viral pathways in their immune response. But interestingly, at day 7, once these patients have been in the hospital and progressed in their ICU stay, basically COVID patients and sepsis patients became transcriptionally indistinguishable from each other, indicating that severe COVID is severe sepsis once they trend towards severe sepsis at the end of their stay. And then lastly, just to tie it all together, what we were also able to look at was whether or not these sepsis endotypes could also be found amongst COVID patients. And indeed, Arjun looked at some data of COVID patients, looking at their gene expression profile using those previous endotypes that he'd already looked at. And he indeed found that he was able to classify patients into their different endotypes. And then also that these endotypes were, again, some of them were associated with high mortality and sepsis severity. And so again, this shows you that there's a real benefit to being able to look at these endotypes in addition to identifying those sepsis patients. So in conclusion, I hope I've given you a flavor of some of this, the idea that characterizing this dysfunctional immune response is crucial to identifying those patients that are at highest risk of developing severe sepsis. This type of approach would be highly complementary to any of the pathogen identification technologies and also all the other tools that are available at a physician's disposal, all these things can be used together to be able to make the most informed decision for a patient. And then I think future diagnostics aimed at understanding and identifying these endotypes could enable personalized treatment for sepsis patients. And then lastly, that rapid sepsis diagnostics are necessary for future pandemics, because as I showed, severe COVID was due to sepsis, and it's highly likely that in every pandemic, those people who die from those infections are actually indeed dying from sepsis. So with that, hopefully I've got a couple of minutes for some questions, but thank you very much. At the back, I guess? I think they need you on the microphones for online. Sure. So to answer your first question, we have not specifically looked at any type of population. We're just looking at patients as they come into the emergency room. Your second one, looking at neonates. So it's a challenge. We have not looked at that as part of this test, and we don't know. You know, sepsis in neonates is going to be different from sepsis in adults, and so other work that is being worked on actually by Andy as well took a similar strategy, but tried to look for gene expression profiles among neonates with sepsis and non-sepsis. And he actually found a difference in gene signature, but it's also challenging to find those types of samples in those studies because they just don't exist. So yeah, so we have not looked at that, but certainly that's something that we'd like to look at in the future.
Biomarkers in acute severe diseases, including infectious diseases, is littered with hundreds, if not thousands, of failed biomarkers. Things that in studies like yours showed good performance, but when tested in humans' disease to see if that actually improved outcomes, didn't. In fact, it's hard to think of a biomarker that has. So my question for you is, are you guys planning, do you have a specific plan to actually do a human trial, a randomized trial, that demonstrates that the use of this improves clinically relevant outcomes for humans with sepsis?
So to answer your question, so we are planning a clinical study. We're planning our pilot study. You're talking specifically about the sepsis test, right? So we are planning on doing a clinical study, and we're going to be doing that. It's going to be it's not going to inform patient decisions, because you have to get the device approved before you can inform patient decisions. So down the road, certainly we would be, once our test is approved, we would be happy to do that type of study to then demonstrate, yes, our test works. Right now, we need to make sure that we get the sensitivity and specificity to demonstrate the device performance. But I think, you know, so it's not a great way of answering your question, I guess, and I certainly appreciate what you're saying, but I think that type of study will eventually happen, and hopefully our test will not be one of the ones that falls by the wayside and actually does do what we say it does. Okay, thank you very much.